So the first presentation in this session is given to us by Eva Clavane, who currently is a master's student at the University of Leeds. And Eva will continue her project as a postgraduate student with Dr. Paul Meakin at the University of Leeds. And the title of Eva's presentation is Investigating the Role of BACE1 in Angiogenesis. So welcome, Eva. If you'd like to unmute your microphone and share your screen and start your presentation.
Can everyone hear me?
Yes, we can hear you.
Okay, thank you. So, hello everyone, thank you for coming. I hope you're all well and safe. My name is Eva Clavane. I'm a master's student at the University of Leeds, and my project is investigating the role of BACE1 in angiogenesis. So the overall aim of my project is to improve the health and longevity of those with diabetes mellitus, which is a chronic metabolic and inflammatory disease which is associated with numerous complications, some of which are evident in the individual's extremities. And not only, and this increases the risk of the individual from getting lower limb amputations. So not only is this a huge devastating socio-economic impact on the individual, but it's a huge burden on our NHS. And these peripheral complications, such as diabetic foot ulcers, are the consequence of insufficient angiogenesis. So, angiogenesis refers to the growth of vessels from pre-existing ones. And in adulthood, it is important in various processes such as pregnancy, menstruation, and wound healing. And briefly, it is a complex and multi-step process, and it consists of endothelial cell differentiation, proliferation, migration, and eventually vessel maturation. And this process is initiated and regulated by various growth factors, including VEGF. But unfortunately, individuals with diabetes have insufficient angiogenesis, and this is due to various factors, some of which I've highlighted here. But my project is going to focus on the increase of BACE1 levels in diabetes and how this reduces the local VEGF levels that cause the insufficient angiogenesis and consequent complications. So BACE1 is an acronym for beta-site amyloid precursor protein cleaving enzyme 1, and it is an aspartic protease with this common aspartic acid motif, but also this unique transmembrane domain, which is important in its activity of predominantly cleaving membrane-bound proteins. And it was identified due to its notorious role of cleaving this amyloid precursor protein to amyloid beta peptides, which contributes to the etiology of Alzheimer's disease. However, BACE1 isn't just limited to cleaving the amyloid precursor protein, and actually has a diverse array of substrates which has previously been unexplored. So these are 4 findings which have influenced my project. So firstly, Kyatel has discovered that BACE1 has a role in developmental angiogenesis, and this is due to the cleavage of the VEGF receptor 1, which is an important receptor in angiogenesis regulation. Additionally, various researchers have found that BACE1 levels and activity are increased in individuals and diabetic models, and this is due to the promoter region of the BACE1 gene having various binding sites for these transcription factors that are associated with cellular stresses, which are abundant in diabetes. Also, this soluble FLT1, which is a truncated product of this cleavage activity, is increased by 2-fold in diabetes. And finally, the local VEGF levels are decreased in diabetic foot ulcers and wounds. So combining these findings, we could hypothesize that BACE1 dependent cleavage of this FLT1 to this soluble decoy anti-angiogenic form reduces VEGF bioavailability and subsequent pro-angiogenic signaling through this VEGF receptor 2. And if this is correct, then BACE1 inhibitors can be repurposed to recover effective angiogenesis in those with diabetes and increased BACE1 levels. So there is some preliminary data which supports my hypothesis. As you can see, when BACE1 is inhibited, this FLT1 is not cleaved. So to begin with, I looked at the effects of BACE1 deficiency in angiogenesis in parameters previously unexplored. And so here I dissected the retinas of 5-day-old pups and stained them with isolectin B4, which fluorescently highlighted the endothelial cells in green. And what I found was that the retinas of the BACE1 deficient mice had a decrease in radial outgrowth, an increase in branching complexity and vasculature area, an increase in filopodia, but surprisingly a decrease in tip cells, but it does mean that my experiment needs to be repeated. But overall, this finding suggests that BACE1 deficient mice have a hyper-sprouting phenotype. So I then investigated this further in vitro using this fibrin gel bead assay. And this consisted of ligating endothelial cells such as HUVEX to cytodex beads and then embedding them in a fibrin clot with growth factors and either a highly specific BACE1 inhibitor, M3, or a relevant control, which is DMSO. And what we found was that in the BACE1 inhibited cells, we had a significant increase in sprouting. However, it had no effect on the sprout elongation. So this supports our in vivo findings. I then wanted to investigate this in a diabetic model. So I produced this doing a hyperglycemia model. So I incubated the fibrin clots in a relevant glucose concentration. But what I found was that the effects of the BACE1 inhibitor were reversed. And this could be because the increase in glucose may increase the BACE1 activity. So then the amount of BACE1 inhibitor that is in there can't counteract the effects. So next, I investigated whether BACE1 inhibition affected VEGF bioavailability and the subsequent signaling through this pro-angiogenic VEGF receptor 2. And I did this by measuring the phosphorylation events of both AKT and eNOS, which are two downstream factors of this receptor. And what I found was that there was a significant increase in phosphorylation of eNOS at basal levels. However, there was no added effect with the VEGF stimulations. So this could potentially mean that the phosphorylation sites are saturated, or that the VEGF stimulations are just not having an effect. And interestingly, the phosphorylation of AKT wasn't significantly different between the two conditions. So it could suggest that this phosphorylation of eNOS is due to different kinases other than AKT. And this is something I'm going to further explore when I'm back in the lab. But because of these previous results of the hypersprouting phenotype and with the ineffectiveness of VEGF stimulations, I diverted my attention to Notch signaling. So Notch is an important receptor in tip cell selection and sprout formation. And when this receptor is activated, it releases an intracellular domain, which then goes on to upregulate genes important in the stalk cell phenotype. And what we found was that in the endothelial cells of the lungs of BACE1 deficient mice, there was a decrease in the Notch intracellular domain, which is actually characteristic of the tit cells. So this could potentially explain the hypersprouting phenotype that we're seeing both in vivo and in vitro. But interestingly, it's also previously been discovered that BACE1 cleaves jagged wood, which is this other notch ligand. However, this can't explain this result, as this ligand is already involved in decreasing notch intracellular domains. So this is something we're really interested in and we're going to further explore in the future. So just to conclude, BACE1 inhibition or deficiency causes a hypersprouting phenotype, and this could have potential therapeutic purposes for individuals with diabetes, as it can encourage the angiogenic potential and then the subsequent perfusion of blood to areas that require it. And then also BACE1 inhibition increases eNOS phosphorylation, but this is more likely due to the BACE1 dependent cleavage of the insulin receptor, which has previously been found, as the insulin receptor is a huge contributor to eNOS activation. And finally, BACE1 inhibition decreases notch signaling, and this is, as I said previously, something we're going to explore further. So I just want to say a huge thank you for everyone who attended and is listening, and a huge thank you to my lab group at the University of Leeds, especially my supervisor, Paul Meakin. A huge thank you to the BMS for not only putting on this great event and giving us these opportunities, but also funding my summer studentship last July, which was great for it actually kick-started my career in academia. And a huge thank you to the British Heart Foundation for funding most of the research that goes on in our lab. So thank you.
Thank you, Ava. That was an excellent presentation, some lovely images and some really interesting data. So does anybody have any questions for Ava? You can either use the chat facility or raise your hand. Just checking whether there's any questions on the chat. Yeah.
I hope everyone's super excited about BACE1.
So we have one question from Breton Barrett. Thank you for a great talk, Ava. I was just wondering, do you know how expression and activity of BACE1 changes with the progression of diabetes with age?
Well, I can imagine that it actually increases with age, and this is evident in Alzheimer's disease, because obviously Alzheimer's disease is predominantly increased with age. So yes, I think the activity in age will increase BACE1 levels.
Okay, another question from Mustafa Samak. Have you investigated the role of BACE1 in HUVEX using different assays, such as tube formation or wound healing? So the more functional in vitro assays.
So these ones focus more on the endothelial cell migration parts rather than the overall. So unfortunately, due to the COVID, I haven't had enough time in the lab to really investigate all these aspects of angiogenesis and the role of BACE1 in them. So this is something I'm definitely going to do when I get back into the lab. And it's something that we have thought about. But currently, no, I haven't used these.
Okay, so something for future studies once you're back in the lab. So question from Giovanni Mann. Does elevated glucose per se affect notch signaling?
So this is not something I'm aware of. It'll be something I will look into, but currently I don't know if glucose affects notch signaling. No. Does anyone else?
Yeah, maybe if anybody has a response to that, they could pop it in the chat. So question from Christopher Kelsall. Are you concerned about the possibility of BACE1 inhibition leading to angiogenic formation of pathologically leaky vessels? It's a good question.
Yeah, so this is something that we will consider and something that we will investigate. So we've currently just been exploring whether these BACE1 inhibitors will just initially just increase the potential of angiogenesis and the actual sprout formation that will hopefully carry on to healthy blood vessels. But obviously it's something we'll look into and something we'll investigate once we come to that stage, once we get to it. Okay,
well, we'll just take the final two questions. How do you know for sure the effector of BACE1 is FLT1, since it has so many other substrates? Could you use some inhibitors of a soluble FLT1 action, for example?
So it was just a hypothesis, and it's something we just need to further investigate. Obviously, we're not sure about this at the moment. We can't certainly say that it is because of soluble FLT1. It was just a hypothesis, and it's something we're going to further look into.
Okay, and finally, have you looked into the effect of glycolysis, and in particular expression of PFKFB3, which is important in sprouting?
No, sorry, I haven't looked at that yet. Obviously, because I'm a master's student and this project has just taken off, we're still in the very early stages, but these are all great suggestions and things we're going to definitely take into consideration when I'm back in the lab, obviously.
Yes, so Ava, perhaps if you have any further thoughts about any of the questions, you'd just pop them into the chat.
Okay, thank you.
So I'd like to thank you once again for an excellent presentation.
Thank you very much.
Okay.